Good morning. This is our first lesson on the excretory system. So the excretory system is one of the more complicated body systems within our body. Now, the excretory system, the first thing that I always ask students when we start our lesson on the excretory system is really, what is it? What does it do? Not a lot of students actually know what the excretory system is referring to, but we can also call it the urinary system. So now that I've written that down, what is the purpose of the excretory system? So the purpose of the excretory system is to create urine. But how is urine created? So this is what we're going to get to talk about today. So we're going to create urine with the help of our kidneys, our bladder, and then some lovely structures that are going to join your kidney and bladder to the toilet. So from analyzing the purpose of the excretory system, is a bowel movement considered part of the excretory system? So when we talk about a bowel movement, that's talking about poop. So if we're talking about the kidneys and the bladder, does poop come out of your bladder? I should hope not. So that means that no, the excretory system is not related to a bowel movement at all. Okay, that is related to the digestive system. So remember in the digestive system, we talked about how our food was broken down and then it is absorbed into the small intestine and then it moves into the large intestine where water is reabsorbed and then it will compact into a nice bowel movement. And then that bowel movement will hopefully go into a toilet, or if you are fancy, you can go outside, dig a hole. Yes, I'm trying to be funny in a video, but it's probably not working. So that being said, we're going to move into the structures of the urinary system, as well as what products are we trying to get rid of in our body with the use of the excretory system or the kidneys and the bladder. So the kidneys are very important to the human body because it has quite a few functions. So number one is removing the waste. That's the primary function of your kidney. And that's what we really want it to do because that's what's going to keep our bodies healthy. And then number two says it's going to balance the blood pH. I am not going to talk a lot about that today. I'm going to do that in tomorrow's lesson because it's a little more complicated than what I'm going to introduce today. The next one is maintaining water balance. Okay, so in our bodies, we need to make sure that we have enough water for everyday function. The majority of our body is made up of water. Roughly 70% of our body is water. So that being said, we need to make sure that we're not getting rid of all of the water that we're intaking. So our kidneys are going to help reabsorb some of that water so we're not having to constantly drink. So moving on to our waste products. So our waste products are talking about the things that we want our body to get rid of. So the first thing that we're going to talk about is deamination. Deamination, sorry, I said it wrong the first time. Deamination is the removal of the amino group from an organic compound. Okay, so when we talked about organic compounds, we were talking about basically the food that we're taking in. So that being said, if we think back to the digestive system, we talked about three different macromolecules and we talked about the breakdown of those three macromolecules. So we talked about a carbohydrate, we talked about a protein, and we talked about a lipid. So when we refer to amino acids, is that actually coming from your carbohydrate intake? Is it coming from your lipid? Or is it coming from your protein intake? Pause for dramatic effect. And here is where I would field questions, but unfortunately I am talking to a computer. So that being said, the macromolecule that contains the amino acids are the proteins. So proteins are broken down into polypeptides. Polypeptides are broken down into dipeptides. And then those dipeptides will eventually be broken down into your amino acid groups. Okay, and then that amino acid group actually has to be broken down further as well. So this process of deamination is occurring in the liver. And this is partially our digestive system process as well as the urinary system. So ammonia is a water-soluble gas. So that means it can dissolve in water. And it is the byproduct of the process of deamination. So this happens to be extremely toxic, which means that we don't want this in our body. So the liver is then going to take two ammonia molecules and then combine them with our carbon dioxide, which is actually, if we think back to the respiratory system, carbon dioxide can actually float in the plasma. Now, if it's floating in the plasma, that means that the ammonia can freely combine with the carbon dioxide. Yes, I focused on the carbon dioxide breaking down or combining with water to make the the carbonic acid, and then breaks down into the hydrogen ion and then the bicarbonate ion. But there is still some CO2 that does stay in the plasma, which is mainly used to get rid of this ammonia so it's not as toxic. So once those two ammonia molecules combine with carbon dioxide, it's going to make a substance to which we refer to as urea. Okay, so I created a reaction form again just to show the reaction that is happening. Again, it's not chemistry based, but if we take our two ammonia molecules and we combine it with one carbon dioxide, we are going to make our urea. And this urea is a component that the kidneys are going to get rid of. So the urea is actually coming from when we consume our proteins, and our livers have, or our liver, so one, our liver has broken down that protein to its simplest component, the ammonia group, or from the amino group, we get the ammonia group. And then it combines with CO2 to make that urea. The next component that our kidneys is trying to get rid of is uric acid. So uric acid is a waste product that's formed by the breakdown of nucleic acids. Nucleic acids is talking about our DNA. Okay, so DNA actually stands for deoxyribonucleic acid. And no, I don't expect you to spell that out. But everything is composed of DNA. Everything that's living, that has life on this planet, has DNA. So that means the food that we're consuming was once living, so it still has to have DNA. That DNA has to then be broken down and converted into uric acid to be excreted by the kidneys. And again, this is a very simplistic version of what's happening. It is more complicated, but if you go into university classes, you may go into more detail about this. Okay, our next page is showing all of the other waste products that are actually getting, or our body is getting rid of. So I talked about ammonia, I talked about urea, and I talked about uric acid. So this is getting rid of by the kidneys, which is the section that we're in. When we look at carbon dioxide is getting rid of, or we're getting rid of it by our lungs. We exhale the CO2. So if you forget how that happens, you need to look back in your respiratory system notes. And then the biopigments and lactic acid, this we would have talked about in the digestive system notes. Okay, we are going to focus today on the yellow. Okay, so now that we know that we need to get rid of ammonia, urea, and uric acid, we need to know how does it get to the kidneys? How are we actually storing it in the bladder, and how does it come into or create the urine that is excreted from our body? So we need to go into the structures of the urinary system. So normally I would have labeling quizzes on the urinary system, but we don't have time for that. So that being said, it is important to know your structures of the urinary system. So here we're going to start with the renal arteries. I do not like the blue, so I'm going to make it back to yellow. So the renal arteries. Now remember back to the circulatory system. The definition of an artery means that it's moving away from the heart. As I was marking some of your circulatory system questions, I noticed that a lot of students put in that arteries contain oxygenated blood. Yes, the majority of your arteries do contain oxygenated blood, except for the pulmonary artery. So we cannot use it as a definition for an artery as it's containing oxygenated blood because of that pulmonary artery. It just happens that the renal artery does have oxygenated blood, and it is bringing oxygen to the kidneys as well as other things. So renal arteries are actually branching from the aorta. Now, we should know from the circulatory system where the aorta is, but I pulled up a picture of the aorta just so that you can see. So we're looking, look at the structure of the heart. Now I'm pointing, I don't know why I'm pointing because you can't see. So look at the center of that man's chest, you can see the heart. Then you see this lovely J curve at the top. That is representing your aorta. It is even labeled the aorta. You can see that the aorta actually goes down the center of the body all the way to your hip section, okay, or to your hips. So your arteries are going to branch from that aorta. So your aorta is not just in your heart. It's going from your heart all the way down your body. And then all arteries are going to branch off of that aorta, which will then branch into arterioles, to capillaries, etc. We are focusing on, in this section of notes, the renal arteries. You can see the two kidneys within this diagram right underneath the man's diaphragm. And you can see the man's diaphragm as like that W shape. That W shape. So your kidneys are located here. So that means my renal artery is here and here, okay? And then it branches into our two kidneys. So going back to our lovely picture here. So this center here, this red, is representing my aorta. And then this blue one is representing my inferior vena cava, which again comes down from, well, technically it's bringing blood back to the heart, so it's going up. So when you have blood coming to the kidneys, you have to have blood going away from the kidneys as well. So that's why you also have your vena cava going down the center of your body, because all veins are going to eventually drain into the vena cava. So you have a big vein, a vena cava, and an aorta going through the center of your body. Okay, so renal arteries branch from the aorta and enter the kidneys. Now, waste is going to be filtered from the blood. Now, this doesn't make sense because we talked about the respiratory system, and the respiratory system, we focused on the waste of carbon dioxide. But remember, that happened in the lungs. We're not in the lungs now. We're going to the kidneys. So The blood that is going to the kidneys in the aorta through the renal arteries does have oxygen and has less CO2, but it hasn't gotten rid of the urea, the uric acid, etc. So that's why the blood needs to go to the kidneys at this point, because we need to get rid of all of that other or all of the other waste products. Okay, now the kidneys are then going to filter all of those products out, and then it's going to send the waste products to the bladder, which means through the ureters. So that means there's a tube that is connecting the kidney to the bladder. There are two urinary sphincters. Now, we talked about a sphincter being a constrictor muscle. So that being said, there are two muscles that are controlling controlling urine output from our body. So the two urinary sphincters are located at the base of the bladder, so at the bottom of the bladder, which releases urine into the urethra. Now, the urethra is the tube that is going to connect the bladder to basically the toilet, where the pee comes out of our body. The first sphincter is involuntary. So involuntary means we don't control it. It's controlled by the brain. So when your bladder is full, that first sphincter is like, oh, I'm done, and it opens or it releases. So that means that that first sphincter, when the bladder's full, is going to let go or release. But the second sphincter is voluntarily controlled. This means we control. So that second sphincter is lower than the first one, which means that we are able to hold in the pee even though the first one has let loose. That being said, it's not good to hold your urine for long periods of time, so you should probably go to the bathroom when you really have to. But that second sphincter you do learn to control during childhood. So, what is the pathway of urine? So, urine is created in the kidney, okay? Now, technically, you have a right kidney and a left kidney because you have two kidneys, okay? But I'm just going to put kidney. From the kidneys, the waste products is going to move down the ureter. It is going to get stored in the bladder, where it will then move into the urethra. If those sphincters have let go, into, hopefully, the toilet like a good boy or girl. So I'm going to add in here that here we have two sphincters. Okay, that are going to control the movement of the urine. Okay, that's the structure. Now we're going to move on to how. How is the urine actually created? This is going to be the bulk of the lesson. This is probably going to be the most challenging, which is great that it's recorded because you can go back and watch it. So the kidney itself has three different layers or structures. We have what we call the cortex, the medulla, and the pelvis. Okay, the cortex is referring to the outside. Anytime we talk about the body and we talk about it in terms of the outside layer, it talks about the cortex. So when we go into Bio 30, in Bio 30, we actually use the word cortex a lot because we move on to other body systems, including the brain. So the brain has a cortex, which means we're talking about the outside layer of the brain. So when we look at the kidney has what we call the renal cortex, but for now we'll just call it the cortex. So renal being the kidney. The medulla is talking about the middle section, and then the pelvis is basically the center. Okay, so our pelvis section is going to be representative of this section here that you see me just highlighted. Okay, so I'm going to highlight the pelvis. The medulla is going to be this section. And then the cortex is going to be this outer layer here. I know I highlighted a bit too much, but you get the point. So the cortex is that outer layer. So the meat, the nephron, this is the functional unit of the kidney. Okay, so your kidney itself has microscopic nephrons. Okay, ooh, I don't like that color. Get rid of it. Okay, so these nephrons are responsible for filtering the various substances from the blood and actually transforming them into urine. So it's not the kidney that's doing it, it's the nephrons found within the kidney. And the fact that they're microscopic means you can't actually see them with the naked eye. So if you were to cut open a kidney, you're not going to see anything but flesh. It almost looks like a cooked chicken breast, okay, because these nephrons are so small. Now, these nephrons are supplied with blood from the afferent arteriole. Okay, remember, it's the blood that we have to filter. So the blood has to come into the kidney somehow, which means that you have to have part of the circulatory system connected to the excretory system. So these afferent arterioles are going to branch off of the renal artery. So we come from the aorta, we go into the renal artery, and then we move into the afferent arteriole. Now notice I left a lot of space down here because writing pathways is probably beneficial. So then we're talking about the blood pathway. We're talking about the substances that, or the substance that has to be filtered. So we're going to start at the aorta. From the aorta, we're going to branch into our renal artery and then move into our afferent arteriole. And remember, an arteriole is just a small artery. These arterioles are going to branch into a capillary bed, which we call in the kidney the glomerulus. Okay, so that means we're going to move in the glomerulus. From the glomerulus, blood is actually going to leave the, not blood, sorry, wastes are actually going to leave the glomerulus. And we're going to talk about this when we look at a nephron. So this is really the filtering site. Okay. Leaving the glomerulus, we will leave through the afferent arteriole. Now, if we look at which arteriole comes first and second, A comes before E. So afferent has to come first, then efferent, which will then connect to the peritubular capillaries. These peritubular capillaries are going to reabsorb things. If you remember back to what a capillary does, a capillary is allowing things to move in and out because it is a very, very thin layer, single cell. So you're allowing things to move in and out. So you have a filtering site at the glomerulus. You have things moving back into the capillary bed. So basically you're reabsorbing here. Reabsorbing. From there, the blood is then going to move into a venule, and then the renal vein, and all veins will drain back into the vena cava. More specifically, this one will be the inferior vena cava, which will then go into the heart, right atrium, right ventricle, etc. So that's the where the blood actually travels. Now, the urine, or the creation of the urine, is known as a fluid pathway. So the fluid pathway is very different. You're not going to have blood go, the full amount of the blood go into the nephron. It's going to be just part of the blood. Okay, so that glomerulus is actually surrounded by a funnel-like structure known as the Bowman's capsule. So all of that filtered stuff, we'll just call it stuff for now, is going to move from the glomerulus into the Bowman's capsule. So our first stop in the fluid pathway is going to be the Bowman's capsule. From the Bowman's capsule, the fluid is then going to move into the proximal tubule, down the loop of Henle, then to the distal tubule, and then to the collecting duct. So we're going to put proximal tubule or proximal tube, okay, loop of Henle, then to the distal tubule and collecting duct. All along this pathway, the nephron, or these different parts of the nephron, proximal tubule, loop of Henle, distal tubule, collecting duct, there's going to be reabsorption of certain things at certain places. And you're going to need to know what is reabsorbed in the proximal tubule, what's reabsorbed in the loop of Henle, etc. So that's what we're going to get into next. So here you can see the kidney. So here is a picture of the kidney. You have blood coming in, blood going out. So this is the unfiltered blood going in, meaning that it has those uric acids, urea, etc. And clean blood, now this will still have carbon dioxide because it's deoxygenated in this scenario, but it won't have the uric acid, urea, etc. This over here, this yellow structure, is representing our nephron. So in our nephron, we have blood coming in. So this means this is our afferent arteriole right here. This moves into our glomerulus here. The glomerulus is where you have the filtering of the blood. Your Bowman's capsule is surrounding this glomerulus, okay? This means this is my proximal tubule because it comes off of the Bowman's capsule. This is my loop of Henle. This is my distal tubule and my collecting duct, okay? Now this is my descending right here. It's descending tube or descending loop of Henle, and then this one's my ascending loop of Henle because it's going up, okay? Now it says highlight the areas of the nephron found in the medulla of the cortex and the pelvis. I'm going to do that later when we do our diagram. Okay, so the 3 steps in the formation of urine. I'm going to talk about these as quick as possible because doing the diagram is going to take quite a bit of time. So we have filtration, reabsorption, secretion. Filtration is one of the easiest ones to talk about. So let's look at filtration first. So blood is going to move into the glomerulus. This is unfiltered blood. We need to get rid of a lot of the waste found within the blood. The glomerulus is acting as a high pressure filter. So the pressure that exists here is actually higher than any of the other capillary beds in the body. So this is great because we know that things are always going to move from high pressure to low pressure. So if I have high pressure in the glomerulus, that means that the Bowman's capsule has to be lower in comparison. So that means that my nutrients, or not my nutrients, my dissolved solutes in water, okay, so dissolved solutes is talking about my waste, are going to pass through, and I'm going to put a point, waste, are going to move through the walls of the glomerulus into the Bowman's capsule. This creates what we refer to as a filtrate because it's not everything in the blood, it's just all of those wastes. So this is like a filtered fluid, very similar to blood plasma. Now, plasma proteins, platelets, and erythrocytes, so that means that the big stuff, Okay, so those plasma proteins, the platelets, and the erythrocytes, they're very big. So that means that they're unable to move through the membrane because they're so large. And if you look at this diagram or this chart below, you can see that the proteins are going to stay in the blood plasma. They're not found in the filtrate. Now, the only reason I'm starting this is so that you know what I'm talking about as I'm talking about it. You don't have to highlight it in your notes package if you don't want to. Then you will notice that you have sodium chloride, glucose, and urea. So sodium chloride, glucose, and urea are all going to be filtered over to the filtrate or to the Bowman's capsule. You'll notice that you have 3 grams per liter in the plasma of the sodium, and then 3 grams per liter of the sodium in the glomerular filtrate. Filtration doesn't mean that it's going to get rid of everything. It is going to try to maintain homeostasis, so it will only move till there is a balance on both sides, okay? Which is why you see on the rest of them that they have equal numbers on both, okay? So your concentration in your glomerulus and your Bowman's capsule is going to be the same, okay? Reabsorption. Reabsorption is one of the more complicated ones, but it's very, very important. We want reabsorption because, number one, we don't want to be peeing all the time. If we don't have reabsorption, that means every ounce of fluid, that's fluid that's in food as well as liquid that you drink, has to come out. So you'd be on the toilet all the time. Number two, we don't want to have to constantly be drinking. Whatever comes out has to come back in. So you don't want to have to be drinking all the time or peeing all the time. So for every 120 mils of fluid that is entering the nephron, 119 of it is reabsorbed. That is a great, great number. Okay, so that means only 1 mil we're getting rid of. So that is a really good percentage being reabsorbed. Now, reabsorption is occurring by both active and passive transport. You should know that from Science 10. Passive transport does not require energy. It happens naturally, so like the filtration part. Whereas active transport is driven by ATP, meaning that it needs energy in order to happen. So, what is being reabsorbed? So, A, B, and C, I've kind of divided it. Carrier molecules are going to carry Na positive and glucose by active transport. Okay, so that means that those positive ions in the glucose, you have to force them out. So that means that we have to use some energy to bring them back into the blood. Now, forcing them out, they can't just go out and be diffused everywhere, okay? They have to go somewhere. So they're going to go back into the blood. Now, since you have a more positive charge in the blood, negatives are going to want to follow, okay? So these negative ions of chloride and the bicarbonate ion are going to move by passive transport because of charged attraction. Since that sodium has already moved out, the chloride is going to want to follow and stick with that sodium. Now, when a threshold level, which is talking about the maximum amount of the substance that can be moved across the nephron until there's no ATP left, is reached, the remainder of the ions will be excreted. So basically, our body's going to reabsorb as much as it can before it runs out of energy, and then we'll excrete the rest. We'll pee the rest out. The concentrated solutes in the blood are going to create an osmotic pressure that is going to push water back into the blood, as well as the proteins that were left in the blood. So the two things that help water go back into the blood or be reabsorbed back into the blood is the fact that you have that osmotic pressure, the other big thing is the proteins. You have big things in the blood. You have no big things in the nephron. So that means that the water is going to want to try to dilute those big things by being drawn back into the blood. And then the last one here is glucose and amino acids. Now, glucose and amino acids are going to attach themselves to carrier molecules. Now, carrier molecules means that you have to use ATP in order for the molecule to change shape and move the substance out. So this is also going to increase the osmotic pressure of the blood, which means that it's going to help draw water back in. So if there's too much glucose, that means that the extra glucose is going to be excreted in the urine. That sounds like a lot, okay? But I'm going to go through it in detail when we do our diagram. The last one is secretion. Secretion is simply meaning that the majority of our nephron has reabsorbed whatever left, we have to get rid of. So it's actually the movement of waste from the blood into the nephron, through active transport and blah, 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 like the previous, but we're getting rid of all of those wastes, okay? Your information, this chart here is from your textbook, okay? I'm going to summarize it when we do our, skip this page for now, it's just another page of what we're going to be drawing out, this lovely page. So this is page 8 in your notes package. If you want to color code like I do, you can pause the video and grab 1, 2, 3, 4, 7 colors. I think it's going to be 7 colors. Okay, I'm going to make this smaller so that we can see everything. Okay, so my first color that I'm going to pick is a gross yellow because I'm only going to use this once, and it's just to show us the separation between the different parts of the kidney. So we have the cortex, the medulla, and the pelvis. The majority of the nephron is actually found in the cortex, which would be the outer layer. The cortex includes the Bowman's capsule, proximal tubule, distal tubule, as well as the top of the collecting duct. So that means I want you to create a line going straight across here so that you can separate this into what we call the cortex. I'm going to run out of room. Do this again. Okay. Make it smaller. Oh, that's colors. I don't know how to make it smaller, guys. Sorry. Okay. I'll just do it this way. Cortex. The layer underneath the cortex is going to be the medulla. So that means under here, I have medulla. Of this bottom corner here, the bottom of the cortex, or sorry, the bottom of the collecting duct. Down here is going to be our pelvis. And remember, the pelvis is the center of the kidney. So where the kidney kind of bends inwards. Okay. Now black. And I'm going to label all of my structures found within the nephron. The structure that looks like, or within my, actually B, we'll start with B. B is going to be my Bowman's capsule. C is going to be my proximal tubule. D is my distal tubule, which makes the structure inside Bowman's capsule my glomerulus. Now, I is part of my loop of Henle, which is my descending loop. G would be my ascending loop. And then H is my loop of Henle. So it would be including the descending and the ascending. Now I think the structure underneath where I wrote medulla is E. So this is my collecting duct. And then F is representing 2, my ureter. So that's all of our labels. Now what I'm going to do is I'm going to divide this diagram into the 3 different processes happening. So I'm going to use a dotted line for this. Okay, so I'm going to put a dotted line, purple, going all the way down here. And then I'm going to put a dotted line exactly where I have my pelvis section. Okay, so this first section, and now I'm going to just write in purple kind of so that you can match it up. So number 1 is filtration. So this happens in the glomerulus going into the Bowman's capsule. Number 2 is reabsorption. And then number 3 happens over here, and this is secretion. Okay. Now I'm just going to make it smaller so that I can write the processes happening in 1, 2, and 3 at the bottom, and then we'll go back to the structure up top. Okay. So page width so that I can actually write meter. So I'm going to use 2 different colors. I'm going to use an orange and I'm going to use a blue. Hopefully. Actually, maybe I'll use, no, use orange. Okay. So, oh, here we go. Line style thickness. Okay, how? How filtration happens is by pressure. So we're moving from high pressure, okay, in the glomerulus to low pressure, which was in the Bowman's capsule. I'm just going to put Bowman's. Okay. We're going to talk about what in a second, just so I don't have to flip back between colors. My reabsorption phase, how and what are going to happen together. So this is going to be a little more complicated. I'll come back to this one. But in number 3, the how is going to be pretty simple. Okay, so how do we actually secrete? Well, it's going from the collecting duct to the ureter, to the bladder, to the urethra, to the toilet. Okay. Now I'm going to use a blue to represent the what. So I'm going to go back to filtration. What is actually being filtered? Okay, so what is going from the glomerulus into the Bowman's capsule? So we have water, we have glucose, we have amino acids. I'm just going to use AA for amino acids. That would be like the uric acid, etc. Urea, Na positive, Cl negative, and hydrogen ions. You do have bicarbonate ions as well, but we're not going to focus on those for Bio 20 as much. Okay, so the how and what we're going to do together with regards to reabsorption. So we're only going to focus on In the case of the reabsorption phase, priority number 1, water. Okay, priority number 2, Na positive, and Cl negative would be the next one. So go back to my orange. So water is reabsorbed by passive transport. So I'm just going to write passive. This is because of the process of osmosis, which is based on concentration. So remember, you're going to have a higher concentration of solutes within the blood, so water is going to be drawn back in. Sodium was by active transport, okay, which means that it requires ATP. And then the last one is passive transport, okay, now due to charged attraction. So listen carefully to this. This is going to be one of your questions on your assignment. So when sodium moves out due to active transport, you're paying for that. You're using ATP to draw it out. And then Cl is going to follow. You're not paying for that. You only pay for the Na. Now you have more sodium and chlorine located within the blood. So those are technically solutes. So now you have more solutes in the blood. Water is going to want to follow to try to dilute them. So you're paying for this Na, and you get Cl minus and water following. Pay for one thing and you get 3. So it's basically 3 for the price of 1. So now that we have that, we're going to go back up to the top and draw in what is being reabsorbed where. So we're going to start in the proximal tubule because nothing is being reabsorbed in the Bowman's capsule because that's only filtration. So I'm going to use green for what's being reabsorbed. So in the proximal tubule, we have glucose being reabsorbed. Now remember, it's not going just anywhere. This is going back into the bloodstream. Okay? Na positive is going to follow. Cl minus is going to follow. Amino acids, AA. And then we also have water, H2O. You could put that at the top if you wanted to. So basically everything's being reabsorbed at the top, okay, in the proximal tubule. Now you have a lot of your solutes that have left. So what's left in your nephron as you're traveling down here, okay, is a lot of water. So what's going to happen is, since most of your solutes are back in the blood, water is going to want to leave to try to dilute that. So in your descending tubule, H2O is going to leave. So in the descending loop, it is not permeable to Na positive, Cl negative. Permeable to water. So basically what that means is only water is allowed to leave. So now as water is leaving, it means that you have a lot of the water going out. So this would be very similar to, I always go back to the coffee pot because a lot of people understand the coffee pot. So in a coffee pot, you have your coffee grounds that are located in the filter, okay? And as water is dripping through that, at the bottom of this loop of Henle, you've had all of the water leave. Okay, so that means that there's no more water or very little water. So that means in the, or at the bottom of the Bowman's capsule, I'm going to point to here, you're going to have a higher solute concentration. Okay, because there's less water. Now, down here on this side, since solute concentration is higher, This means we want to reabsorb some of that. Okay, so Na positive and Cl negative are going to move out. Okay, so we can draw in our diagram, Na positive is leaving and Cl negative is leaving. And you can put more Nas and more Cls throughout that. Now, this also means that the ascending loop is permeable to Na positive and Cl negative, but not water. Okay, so the ascending permeable to Na positive, Cl negative, not H2O. So now you have more of your solutes being reabsorbed. So at the top, in our distal tubule here, you would have had a lot of your solutes leave. So what that means is there's less solute and more water. So the solutes are always the Na, the Cl, the glucose, etc. So this means, therefore, H2O moves out. So now we can draw in our H2O leaving. The descendant for the collecting duct is also going to reabsorb water. You'll notice that there's always a lot of reabsorption of water because we need to reabsorb that water to maintain efficiency within our body. Okay. Now the collecting duct, I'll see if I have enough room here. This is increasing concentration of solute. When you have square brackets, it means concentration in medulla causes H2O to move out. So the lower you are in the medulla, the higher the concentration of the solute, okay? Because most of the solute is actually leaving near the bottom of the medulla, closer to the pelvis. I realize that this is a lot, but that is how the nephron works. There is 3 practice quizzes, but you can just call them practice assignments. You can practice doing this. You need to do quite a few practice sheets in order to get these concepts hammered into your brain. Good luck.